So in today's lesson, we're going to be looking at the next stage of cellular respiration, aerobic cellular respiration that is, by looking at the electron transport chain. So during glycolysis and the Krebs cycle, during those processes, along the way we saw that there were some ATP molecules that were produced, but not very many. And so, and that was done via the process of substrate level phosphorylation. And you'll remember at the very beginning of this unit, we discussed that there were two types of phosphorylation: substrate level and what was the other one? Oxidative phosphorylation. And that somehow our electron carriers NADH and FADH2 would be really important for that. So during glycolysis and the Krebs cycle, we produce some of those electron carriers in varying numbers, and now is where we're going to see them actually play an active role. We're going to see, well, why were they so critical along the way? So let's take a look. All right, so there's a lot to look at here now that I've opened up this screen. So let's just take it one step at a time. I want to orient you with respect to this diagram here so you have some idea about what you might be looking at. So in terms of cues, well, we can see here that I have, a phospholipid bilayer. And so let's look at some of the labels. Well, it appears that on the bottom half of this is the mitochondrial matrix. So that's the inner part of the mitochondrion. And then over here on the upper portion, we're looking at the intermembrane space. So I'm just going to draw a quick little picture here. So, imagine I have a mitochondrion here. And the cristae, right? And so, well, what are we looking at? We're looking at what is happening across this membrane here. So imagine that we've zoomed into a zone of that, or maybe this would be like a better zone because it's horizontal. Anyway, and so we've zoomed into that section here, and that's what we're looking at. And so it's important for us to note that, we're looking at the directionality of how particles are moving, from, maybe across this membrane in one direction or another. So that's going to be important as we move forward. And there's some other things that you should notice here. You should notice that we have some integral proteins or integral molecules of some description, and they're going to also play a part in this electron transport chain as well. And you'll notice that, we seem to be starting with NADH, one of our electron carriers. FADH2 will come in as well, and we're ending off with water, and that's going to be important, and we will get to it. Some other key molecules are these H+ ions, these protons. Okay, so let's begin with our conversation. So when I say that the electron transport chain, shortened to ETC, components are a series of compounds, mainly proteins, associated with the inner mitochondrial membrane, hopefully that makes more sense now that we've had this conversation about the diagram. Now we can also say that in terms of these proteins, that they're arranged in order of increasing electronegativity. So you'll remember that electronegativity refers to an atom's ability to grab electrons and pull them towards itself, right? So it's an electron grabbing ability, if you will. And so, what we can say here is that these are ordered in order of increasing ability to attract electrons. So let me just imagine that this little red dot here on your screen is an electron held by, well, I'm going to say even NADH, right? So NADH will be, let me add it in here to this diagram. So NADH, and imagine that NADH, imagine that NADH has its little electron, right? And it's an electron carrier, and so it's carrying electrons. And so it arrives at the electron transport chain, right? It's ready to be undergo oxidative phosphorylation. Okay, remember that it was reduced earlier. NAD+ was reduced to NADH, and now we're going to oxidize it. So oxidation, you'll remember, means losing electrons. Let me just remind you of that. Losing electrons is oxidation, and gaining electrons is reduction. So NADH will have its little electron and it's holding on to it, but it can't hold on to it very long at the beginning of the electron transport chain. Why? Because NADH dehydrogenase is more electronegative and can easily grab its electron, right? So in any process, so what's going to happen along this electron transport chain is we're going to shuffle that electron to increasingly more electronegative compounds. Okay, so for instance, NADH dehydrogenase is able to oxidize NADH and grab its electron, but then it is quickly oxidized itself as that electron is shuffled along the line. So earlier in the unit, we talked about coupled redox reactions where one substance in the chain is oxidized and then reduced. This is where this plays out. Okay, so it's a series of oxidations and then reductions as those electrons get shuffled. Okay, so let's write some of this down so you have time to process some of that and hopefully internalize some of those ideas. So each compound in that electron transport chain is alternately reduced and then oxidized. So when we say reduced and then oxidized, remember that we're beginning this process with I'm going to have to write that in a different color, NADH dehydrogenase. That's what we're saying is being reduced first, right? As NADH is oxidized. Okay, so those are coupled redox reactions, as mentioned. So, well, where are those initial electrons that are being gained by NADH dehydrogenase coming from? Well, they are coming from NADH and FADH2. Okay, so now I need to talk about the next piece of this, which is that what is the point of those electrons being shuffled? Why do we care about that? Well, you'll remember that when a molecule or an atom really wants an electron badly, then it is likely to be unstable. So we would say that we have not only molecules or proteins here of increasing electronegativity, but also increasing instability. So as soon as this NADH dehydrogenase grabs the electron that it wants from NADH, it becomes more stable. So, and we can think of this as it becomes less stressed, right? So what happens when a molecule becomes less stressed? Well, it's going to release energy. And so, we're going to harness that energy. The whole process of cellular respiration is to harness energy from the chemical bonds in glucose. So when NADH dehydrogenase accepts the electrons, it is going to release some energy, and we're going to use that energy to take any hydrogen ions or proteins that are in the mitochondrial matrix, and we're going to pump them out into the intermembrane space. That is what that energy is going to be used for. So as the electron gets shuffled to the next substance in this electron transport chain, the next species, electron or energy is going to be released. So as an electron is added to cytochrome B-C1 complex, it also becomes more stable and releases energy. And what is that energy going to be used for? Again, to pump those protons into the intermembrane space. Okay, so that is happening as we move from NADH dehydrogenase, cytochrome B C1 complex, and then finally our cytochrome oxidase complex. In the middle here we have ubiquinone and cytochrome C, which are just cofactors that are involved in the process. And so, I think we're ready to write some stuff down now. So there it is. as electrons are shuttled along the chain, they become increasingly stable, and that allows them to release energy. That energy is used to pump hydrogen ions or protons from the mitochondrial matrix to the intermembrane space, and that happens via those 3 protein complexes that we spoke of: NADH dehydrogenase, cytochrome B-C1 complex, and cytochrome oxidase complex. And so those in that order. Well, now it's time to talk about, well, what happens to those 2 electrons that NADH gives up, right? Where do they end up? They have to end up somewhere. And so let's look. Well, you'll remember that the overall reaction, where am I going to write this out? The overall reaction for cellular respiration is that we're going to have our glucose, our C6H12O6. We're going to have oxygen, 6 of them, in fact, and we're going to produce 6 CO2s and 6 H2Os, right? Well, we already know what has become of our glucose molecule, right? During glycolysis, it was broken down into 2-pyruvate. During pyruvate oxidation, it was broken down further into acetyl coenzyme A. And then in the Krebs cycle, it was, those acetyl coenzyme A's were decarboxylated to produce CO2. So there's our CO2 count there, right? From pyruvate oxidation and from the Krebs cycle, right? But what about those oxygens, right? Glycolysis is an anaerobic process. So where do those oxygen come in? Well, here is where they come in. So you'll notice here that at the very end of our electron transport chain, those electrons are grabbed by some hydrogen ions. We have hydrogen ions, and together those hydrogen ions will combine with oxygen to produce water. So the electrons will neutralize those 2 protons. 2 electrons plus 2 positive ions will neutralize. Combining with the oxygen, I end up with water molecules. So we would say that the final electron acceptor in the electron transport chain is the hydrogen ion. And we would say that the final product of the electron transport chain is water. So let's get that last little bit down. Okay, so the 2 electrons will combine, at the end of the electron transport chain, will combine with 2 hydrogen ions, 2 protons in the matrix that are just there, and a half of an oxygen. Now we know from chemistry that, we're just looking at mole ratios here. It's not actually half of an oxygen molecule that exists really for any real time, and that will form water. So it may have occurred to you that we've only really talked about NADH here. We haven't talked about FADH2, our other important electron carrier. So now's the time where I want to talk about that. So if we were to compare NADH versus FADH2, we would say that FADH2 is a little bit more electronegative than NADH. In fact, so much so that FADH2 will not give its electron up to NADH dehydrogenase, that first protein complex in the electron transport chain. So what that means is that FADH2 in the electron transport chain isn't actually giving up its electron at that first stage. So what does that mean? Well, NADH will release its electrons to the first component, that NADH dehydrogenase, but FADH2 will only release it to that second component, our cytochrome B-C1 complex. Okay, and so, that has some consequences. I'm going to pull out a picture here so you'll see this in real time. Actually, you know what? I think I'm going to just scale back to the last diagram. Let me pull that up. There, that's better. And so, if we have, I'm going to use a different color here. If we have FADH2 only coming in here at that second stage, then how many protons is it going to be able to pump into that matrix? Well, FADH is able to go through all three of these components of that electron transport chain, which means that it's pumping out 1, 2, 3 protons for every NADH molecule. Whereas FADH2 is starting right here at that second complex. And so it's only going to be able to successfully pump out 1, 2 protons per FADH molecule. Okay, I'm going to go back to the last diagram. Right? So, again, FADH2 can only start its journey in the electron transport chain at that second component, our cytochrome B-C1 complex. So that has some consequences. NADH is only going to be able to generate 3 protons, but FADH is going to be able to generate 2 as a result. Now, it turns out that there's a direct correlation between the number of protons pumped into that intermembrane space and the number of ATP that are produced as a result. There's a 1-to-1 ratio. So as a result, NADH is going to be able to produce 3 ATPs via chemiosmosis, which we will get to in tomorrow's lesson or the next lesson, while FADH2 is only going to be able to produce 2 ATP, and of course that is per FADH2 molecule. Okay, so now what we've also done that we haven't really talked about yet in this process is that once NADH at the beginning of the electron transport chain is oxidized back to NAD+, it's going to be ready to go ahead and return to the beginning of glycolysis, the beginning of cellular respiration, and get used again. Right? So, in other words, it's recycled. And so, again, when FADH2 enters the second stage of the electron transport chain, it's converted back to FAD and then can go back and be recycled again during the Krebs cycle. So, one of the utilities of the electron transport chain is not only to help to fuel the electron transport chain and thus produce ATP by oxidative phosphorylation. Its function is also to regenerate our NAD plus and FAD, our electron carriers, so that they can be recycled. Okay, so here's our overview here, right? So, we have NADH from glycolysis produced that are going to travel across the inner mitochondrial membrane into the matrix. There we're going to produce more of it in the Krebs cycle and along with some FADH2. Those are sort of lying in wait to be used in the electron transport chain where both NADH and FADH2 are used. NADH begins the electron transport chain at that first complex, NADH dehydrogenase. FADH2, being a little bit more electronegative, begins at the second species in the electron transport chain. As a result, NADH is able to pump 1, 2, 3 hydrogens across the membrane into the intermembrane space from the matrix, producing 3 ATP as a result. And FADH2, because it's more electronegative, is only going to be able to pump out 1, 2 protons from the matrix into the intermembrane space, ready to produce 2 ATP. So that is it for the electron transport chain. And there's a lot that we learned in this 20-minute video, roughly. So feel free to watch it again and to reach out to me with any questions or points of clarification if needed via Google Classroom.